the meeting. So now we are live. So we have our first speaker with us, Dr. Aditya Kaushik. He is a medical doctor with more than 20 years of experience managing medical affairs, regulatory affairs in pharmaceutical industry. Presently, he is a senior research officer and regulatory officer at CDSA Trustee under Department of Biotechnology, Ministry of Science and Technology, Government of India. So I welcome you, sir, and during the session, the viewers can send their queries into a chat box. So once the session will finish, we will take the queries through the chat box. So hand over to Dr. Aditya Kaushik. Sir, please, you can
Yeah, thank you, Dr. Amit, for your kind introduction.
take.
Honorable Professor Y.K. Gupta, sir, Shri Praveen Garg, Chairman, ISF College of Pharmacy, Dr. G.D. Gupta, Dr. R.K. Narang, and all of ISF family, along with learned fellow speakers, and all the participants who have connected online into this session. आप सभी को मेरा सादर नमस्कार. In this session, what I'm going to share is a kind of overview of the topic, which is repurposing of drugs in COVID-19. And when I say overview, I borrow the words from Professor Y.K. Gupta, sir, that so much is happening in the scientific field and so much is yet to happen to find a preventive vaccine or a treatment for COVID-19. And what I'm presenting today is just a drop from that ocean. So I would encourage the participants to kindly go into details of these presentations, the articles that we are presenting here, and find out more details about the trials that are happening on drugs and the vaccines that are being researched for prevention. I'm also thankful to all the scientists who have contributed to the science behind the drug which are being developed for COVID, and grateful to for their contents that they have published so far in various journals. Context, I'll start with a brief background about this topic. 30th December 2019. WHO office in China, Beijing, received information about an outbreak of pneumonia-like illness, which is of unknown origin. 5th January 2020. Scientists in the Chinese virology lab decode the pathogen and found it to be a coronavirus. 11th Jan 2020. The genomic sequence of the pathogen, that is the coronavirus, was shared with the scientific community across the globe. 30th January 2020, WHO notifies this illness to be a global public health emergency. Now, as the coronavirus spread across the globe, scientists start a hunt for a preventive vaccine and a treatment or a cure for the illness. In a month's time, about 40 clinical trials get registered at the clinical trial registry, and these are few of them. This is a snapshot of the clinical trial registry, wherein you can see trials register for hydroxychloroquine, chloroquine, remdesivir, abidol, lopinavir, ritonavir combination, and others.
We will complete.
So, with this background, what I wanted to share is that you may remember that finding a new treatment for a new illness takes years of preclinical testing, which is then followed by human trials. But in this case, the human trials began within 1 month of world finding this new illness happening to the human beings. You must be wondering on how come when a drug takes years of preclinical research to reach the human trial phase is taken so quickly for human trials or for human use. And the answer for this question lies in drug repurposing, which is the topic of my presentation. Drug repurposing is also called as drug repositioning. And to call it in simple words, it's the old drugs when it is subjected to new uses. So old drugs, when tested for new uses, is simply called as drug repurposing. So the next question comes, what does drug repurposing involve? It involves the clinical investigation of existing, so this is important, existing approved drugs for new therapeutic purposes, indication, and diseases. Because if the drug is not approved, it has to have data on preclinical animal testing data, in vitro laboratory data, and then only it goes to the clinical trials and then gets approval from the regulators. So in case of repurposing, it involves the clinical investigation of existing approved drugs for new therapeutic purposes, indications, or diseases. And it's a universal strategy which is followed to find treatment for rare and neglected diseases, wherein it is very difficult to develop new molecules and new drugs. So how does this happen? Drug repurposing actually capitalizes on few facts. And they are, first, approved drugs or abundant compounds, means which were originally developed, but they did not find use further and they were shelved. It also recapitalizes on the information which is available on drug chemistry, its potential toxicity, pharmacology, its formulation, and in-dose. And it capitalizes on the important point, which is these drugs are already tested in some way in humans previously. So what are the advantages of drug repurposing? The first advantage that it offers is it reduces the time and cost of the medicine to reach market. And the reason is because it negates or avoids the need for preclinical development and phase 1, and in some cases, phase 2 clinical trials as well. The second advantage is that existing pharmaceutical supply chains of manufacturing and scaling up the production are used in such kind of repurposing. The third advantage that we see is the known possibility of combining with other drugs could allow more effective treatment. And you may be hearing from the information in the publications that trials are happening on combination of 2 or more drugs in a pursuit to find treatment for COVID-19. Drugs that are being tested, their pharmacology is known, their toxicity is known, the drug interactions are known, so it becomes easy for them for clinical testing. Repositioning also facilitates the discovery of new mechanism of action for old drugs and new classes of medicines. So the next step is the challenges. What are the challenges in drug repurposing or repositioning? Firstly, the dosage required for the treatment of the new illness or the novel disease usually differs or may differ from that of its original target diseases. So you tested a dose in an illness previously, but that dose might not work in the new illness that you are about to test. So this brings in a lot of challenge for the scientists and for the investigators to find out the right dose should be tested quickly for the new challenge. And for this reason, investigators may have to begin from phase 1, then to phase 2 and phase 3, which essentially strips the drug repositioning of its advantage. The second challenge that we see, all these molecules are developed through some labs or through pharmaceutical industry, and there are often patent issues which brings in complexity for further research on these molecules. So you need to have rights, you need to have adequate permission to further research those drugs from the original company. So what are the few examples from past in drug repurposing? So you must have heard of the drug thalidomide, which was originally introduced in 1957 in Germany for its sedative effect, and then was subsequently used to treat morning sickness in pregnant ladies, but was then shelved or was withdrawn from the market because of the adverse events it caused, the birth defects it caused in the newborn babies. And this medicine has now been revived and is being repurposed for the indication, which is leprosy and multiple myeloma. Sildenafil is another drug which was repurposed for pulmonary hypertension. The original indication was erectile dysfunction. There are antifungal agents, clotrimazole, ketoconazole, which are being investigated in anti-trypanosome therapy. So you must be wondering on how does repurposing fits with the new virus or the COVID-19 illness. So drug repurposing in case of antiviral drugs. So what are the advantages that we have been able to use in repurposing some of the drugs and sending them into the clinical trial phases so quickly for their antiviral property against COVID-19 or coronavirus? From the past experiences or investigations, we know that few antiviral drugs have shown to have activity against broad spectrum of viruses, including coronaviruses. And this comes very handy when the scientists decide to repurpose any drug. These drugs are known to target common enzymes, molecular pathways, and proteins across a variety of viruses, which makes it a bit easy for the scientist to repurpose the drugs to target some molecular pathway or enzyme or a protein in the novel viruses. Also, we have in our hand in vitro activity, which is also estimated through effective inhibitory concentration, EC50 or EC90 against viruses. And we also know the plasma concentration which these drugs achieve in humans through our previous investigations. So all these information brings an advantage for the scientist to repurpose these antiviral drugs in cases of novel viral illnesses. And of course, these drugs have known pharmacokinetic and pharmacodynamic properties. Their mechanism of action is known. Their side effect profile is known, and dosing regimes are also known. Look at the effective inhibitory concentration in laboratory, and you can also find out the minimum plasma concentration which these drugs achieve to finally pursue the clinical trials on these drugs. And therefore, it becomes reasonable to clinically test them in new infections, public health emergencies, just like the coronavirus emergency, where there is no other therapeutic drug available. Some examples of repurposed drugs in COVID-19. You might have already heard of these names: hydroxychloroquine, remdesivir, favipiravir, lopinavir, and ritonavir combination, interferons, interleukin-6 inhibitors, tocilizumab, and there are many others which are being tested. Now, uh, there are so many details, and I'm afraid I may not be able to provide information or details on all these drugs during this short session, and therefore I would suggest that you go back to your desk and find out more information about the drugs that are being tested, how they act, what are the clinical trial information reports which have been published so far. So in next few slides, I will only pick a couple of examples from this list to share with you the information. And I'll try to be simple. I will not use high statistical jargons for you to understand. Anybody interested for those statistical significances or more complex analysis, I would encourage that they visit the publication in original and find out more from there. So keeping in view the larger participants' interest, I will just touch upon very briefly on few key points on these drugs. So the first is very well known and everybody knows about this, hydroxychloroquine, which is in short called as HCQ. It's an anti-malarial drug, and it has been found to have in vitro antiviral activity as well. Some studies suggest that it interferes with the virus entry into host cells by interfering with the ACE2 receptor attachment of the coronavirus spike proteins. Studies also suggest that it interferes with the protein synthesis via glycosylation inhibition. And based on its these antiviral activities, it is being tested in clinical trials in immunodeficiency syndrome or HIV. So what is the evidence so far about HCQ in COVID-19? We would say it's not conclusive as yet, either way, positive or negative. So I have used the term inconclusive. Evidence from a few studies or a couple of studies have been favorable for this drug, but these are all small sample size studies. There are other small studies which have shown a negative effect on disease outcome. One such study, which is a positive study about hydroxychloroquine, was published in International Journal of Antimicrobial Agent. And this trial essentially tested hydroxychloroquine alone with hydroxychloroquine plus azithromycin versus a placebo or no drug, which included only standard of care. And this was published online on 20th March 2020. So the key results from this trial, as I said, the trial suggested that 100% patients treated with HCQ in combination with azithromycin were cleared of the virus, that is coronavirus, and 57% in HCQ alone arm were cleared of virus, whereas only 12.5% in control arm were cleared of virus. The authors note the limitation as a small sample size because there were around 46 patients that were included in this study. And as Professor Y.K. Gupta said, that it is very difficult to do a proper sample size studies in cases of emergency, and you need to have some adaptive trials wherein you can be flexible with the sample sizes and you can look at the data. You can do interim analysis to find out how the drug is behaving. So whatever works in this emergency would be very useful at this stage, unless we have a sure shot cure or therapy against COVID-19. There is another trial which is sponsored by University of Oxford. It's a prophylactic study which will be investigating chloroquine as a preventive medicine in COVID-19 This study is planned, and yesterday there was a news item which suggested that the recruitment in the study has started across the globe, which includes Asia, Europe, and other countries. The trial is registered at clinicaltrials.gov. I move to the next important medicine which is currently being tested, and which is remdesivir. Remdesivir was originally created or developed by Gilead Sciences USA in 2009 for hepatitis C. It was later repurposed and tested as a potential treatment for Ebola virus disease somewhere in 2016. And when this outbreak of coronavirus happened, the company started in vitro examination in animal models in January itself to find out that this drug has some laboratory efficacy, in vitro efficacy against SARS-CoV-2 also. And this was based on the result that they found out while they tested this drug against severe acute respiratory syndrome, SARS-CoV, and the Middle East respiratory syndrome, or MERS, which you would have already heard in various articles and various publications. So what is remdesivir? Remdesivir is a nucleotide analog. And what is a nucleotide? It's a basic unit of your DNA, which comprises of a nitrogenous base plus a ribose or deoxyribose sugar and a phosphate group. So all these three combine together to form a nucleotide. So it's a look-alike of the real nucleotide which is present in your body, and it may not have all the properties of that nucleotide, which is important for DNA or RNA construction or replication. And therefore it is called as an analog. It's a copy, but it doesn't have all the qualities. And this is why it is useful in preventing the virus, as I will explain you in the next slides. It inhibits RNA-dependent RNA polymerase enzyme, which is used by the virus to replicate its mother RNA. Coronavirus is an RNA virus. So RNA-dependent RNA polymerase is helpful to the coronavirus, as it helps in replication process in making copies of the original RNA. And all these copies get enclosed in the viral envelope proteins, and they come out of the infected cell and infect other cells, and this is how it spreads. Remdesivir has been shown to have in vitro efficacy against COVID-19 in Vero E6 cells, and the EC90 has been found to be this value. And you can look at the details about this research on this article published online. So how does a nucleotide analog or remdesivir act? So these are the steps. It's a prodrug, which means it's not active in its original form. It reaches inside the body and it reaches the host cell inside the cell, which is infected with the virus or the coronavirus. And there it gets converted into its active form, called as or numbered as GS441524. The next step is this active form gets phosphorylated or further activated to its active metabolite, which is a nucleoside analog. So initially I explained nucleotide. Nucleotide is a nucleotide is a basic unit of DNA or RNA, and nucleotide contains nitrogenous base, adenine, guanine, cytosine, thymine, or uracil, along with a ribose or deoxyribose sugar, plus a phosphate group. In case of RNA, what happens in this situation is the active drug gets converted into active metabolite, which is a nucleoside analog, and it is very closer to the adenine, which is present as a nitrogenous base. And this active metabolite is also called as C-adenosine. C-adenosine. Now, again, this is an analog, this is a copy, it is not original. So it will have some properties, but not all the properties of the natural nucleoside which is present in your cells and which is used by the coronavirus RNA to build its further copies. This active metabolite in the third step gets incorporated into the copy of viral RNA strand which is under replication in place of the actual or the real nucleoside. So it gets attached there, it's a fake nucleoside, it gets attached there, and this is how it interferes with the RNA-dependent RNA polymerase action, which ultimately leads to the termination of the viral RNA production, and the virus is unable to make copies of its original RNA. And this is how the antiviral activity of remdesivir helps in clearing the virus from the host. This is further explained in this picture. On the left side of your screen, you can recognize this picture because you must have seen it many times. It's a coronavirus, and there are many spike proteins on the envelope. The coronavirus approaches the host cell once inside the body through your nasal or respiratory passages. It reaches the cells, and on the cell there are receptors called as ACE2 receptors, wherein this coronavirus sits on this receptor, and it goes inside the host cell through the process of endocytosis. Once inside the host cell, it releases its RNA, and the comb-like structure that you are seeing further right on your screen with colored tooths. I've just used this as a depiction. It's not the actual structure of RNA, it's just a depiction, it's an animation. So viral RNA, this is viral RNA, and all these color pricks are the nucleotide bases, or adenine, guanine, etc. And this viral RNA, using the ribosome inside the cells, produces protein called as or enzyme called as RNA-dependent RNA polymerase, which is shown here in green color. And this RNA-dependent RNA polymerase actually then starts making copies of the original viral RNA. So this is a kind of general description how it happens inside the cell. Now, further down below on the left side on your screen is shown remdesivir, its chemical structure. So when the person is given remdesivir, the remdesivir molecules reaches inside the cells and gets converted into active GS441524, which mimics a nucleoside, which RNA-dependent RNA polymerase is unable to recognize at the moment, and this nucleoside gets incorporated into the replicating RNA, and this is shown here by a red box, rectangular box in the RNA structure, and this leads to further inhibition of nucleoside or nucleotides addition into viral RNA, and it terminates the viral RNA replication, leading to viral clearance. So with this background on mechanism of action, let's have a look at remdesivir clinical trial, which has been published so far in COVID-19. You will find an article which was published in Lancet journal on April 29th. And the title is Remdesivir in Adults with Severe COVID-19, a randomized double-blind placebo-controlled multi-center trial. This trial happened in China in February and March, and the results are published here, which I'll share very briefly. So this trial originally planned about 453 patients to be included. but finally could only enroll around 237 patients. And the reason given by the investigators is that the pandemic or the viral infection, coronavirus infection, got under control in China, and they were not able to find appropriate patients for further inclusion in this study. They included the patients which had up to 12 days of symptoms onset from the illness, and the two major endpoints they tested in this trial are given in the table. The first is time to recovery, and the second is mortality. And they compared a remdesivir arm with a placebo arm or standard of care arm. And what they found in the trial was that the time to recovery in remdesivir group was 21 days from onset of treatment, whereas in control group it was 23 days. Mortality was recorded as 14% in remdesivir group versus 13% in control group. And both these findings are not statistically significant, means the trial could not show benefit with remdesivir in patients suffering from severe COVID. So severe COVID is important here because in next few slides I'll share why it is important to differentiate between severe COVID patients and patients having mild to moderate illness, because there are articles and research which have happened suggesting improvement if the patients are enrolled early on in the treatment as compared to them waiting for getting into severe illness or severe disease zone. Authors' inference in this study was there is no statistically significant benefit with remdesivir beyond those of standard care of treatment, and the limitation they suggested that the trial sample size was inadequate as the outbreak was brought under control and patient enrollment stopped in the trial. The author suggests that further research is needed to be more conclusive about remdesivir. So this is the trial which did not observe a benefit from remdesivir. So this does not mean that remdesivir is not effective. It's just that this trial could not find benefit, and the limitations are already known, and you may go to the original article to find out more details about this study. Surprisingly, on the same day, that is on 29th April, when this study, the study done in China on remdesivir was published, National Institute of Health under United States government, they published a report on the clinical trial result, which said that the trial shows remdesivir accelerates recovery from advanced COVID-19. Now, this trial final report is yet to be published. This was a prelim or interim analysis based report, which was published by NIH for information of all concerned. So how this study was done, I'll just share a brief about this study as well. This study was called as adaptive study because, as Professor Y.K. Gupta has already explained to you, that there is a flexibility in continuing the trial, in analyzing the results or stopping the trial midway. So based on the outcome that an independent body, which is called as Drug Safety or Data Safety Monitoring Board, decides. This trial was originally planned to include 1,063 patients, and this number was based on the power analysis, which is important in statistics. The endpoints, again, in this study remains the same as of the previous study, and this helps in kind of comparing, though it's not scientifically rational to compare two different studies. but it helps in understanding the difference in the outcomes in the two trials. The first endpoint was time to recovery, and the second is mortality. Now, the Data Safety Monitoring Board was constituted, which is an independent body, independent of the clinical trial team. And the role of DSMB, or Data Safety Monitoring Board, is to review the efficacy and safety data while the clinical trial is on. And based on the information they receive, they decide if the trial should continue or it should stop. Most often what happens is trial is stopped midway if the efficacy is found to be stronger, or if the safety of the drug is an issue. Now, if the efficacy is found to be stronger in the interim analysis, which means essentially the trial is able to achieve the primary endpoint which was defined, then the trial is stopped because it becomes unethical to continue giving placebo to the placebo arm when you have already found out that the drug is working in patients, and therefore it becomes unethical if you further continue the trial. And this is the reason that, based on the primary endpoint, which is time to recovery, this trial was stopped midway as it reached the statistical significant value. So let's see the results. Time to recovery in remdesivir group was 11 days versus control, which is 15 days, and is statistically significant. Mortality was 8% in the remdesivir group, and in control arm it was 11.3%. So there was a gain of 3%, and this is a huge gain when you don't have any therapy or treatment and you are in the midst of an emergency, health emergency. Even a life of one patient saves matters a lot, and there is no statistical analysis that could better it. So this result was based on this. And the other thing is the difference in the mortality was not though statistically significant, but absolute numbers are significant. It is assumed that even the mortality would have gained a significant difference had this trial been allowed to continue to its final stage, including all 1,063 patients. So based on this clinical trial report, and as I said, the final report is yet to be published, and that report will have all the details about the clinical trial, on how it was conducted, the methods, everything about that. Based on this trial result in US, remdesivir received approval. On 1st May 2020, the US Food and Drug Administration granted Gilead emergency use authorization for remdesivir to be distributed and used by licensed healthcare providers to treat adults and children hospitalized with severe COVID-19. So this was an important step in our pursuit to find a cure or a therapeutic option against COVID-19. This trial is being, the trial on remdesivir are happening in other parts of the world as well. And hopefully we will find some positive information, more positive information coming out of those trials. Briefly, I will touch upon another clinical trial on a triple drug combination, which includes interferon beta 1b plus lopinavir and ritonavir combination, along with ribavirin. This trial was published, and this study was conducted in Hong Kong. So looking at the design in brief, it included about 127 patients, and the symptom duration at the time of inclusion was 14 days or less. The endpoints in this trial were time to resolution of symptoms, length of hospital stay, and mortality. And this triple combination was compared against control. So time to resolution, the primary endpoint of symptoms was 4 days in the triple combination group versus 8 days in the control arm. Length of hospital stay was 9 days in the triple combination therapy group compared to 14.5 days in the control arm. And there was no mortality in this studies. Early treatment with a triple combination is appropriate because, and this is a very good and scientific reason, because the viral load of SARS-CoV-2, that is COVID-19, peaks at around the time of symptom onset. So, and this is a huge difference from the SARS or MERS, where a viral load used to peak at around 7 to 10 days after symptom onset. So a patient who has just got infection and are having mild symptoms in COVID-19 will have a viral load which is at peak, though he may not have symptoms which are of severe concern, and therefore he may wait for some more days for symptom to worsen before approaching a medical doctor or hospital. So this is a very important finding from the study, that early treatment, if initiated, can save a lot more patients with these kind of drugs or combination drugs. I hope, Dr. Amit, I can take 5 minutes more for a couple of slides. The next combination which is being tested is lopinavir, ritonavir, and ribavirin. Or again, the triple combination therapy had all these three drugs when it was tested. Lopinavir is always given in combination with ritonavir, and the reason I'll share, both are protease inhibitor, but the activity of lopinavir is more as compared to ritonavir. Lopinavir given alone has insufficient bioavailability, so you have to give a lot more drug for achieving plasma concentration which are therapeutic. And this is the reason ritonavir is given in combo with lopinavir, because ritonavir is a potent inhibitor of intestinal and hepatic cytochrome 453A4, which inhibits the metabolism of lopinavir and help in enhancing the plasma concentration. This combination had previously been tested in HIV and AIDS, and ribavirin, again, oral nucleotide analog, means it's not an original nucleoside, it mimics a nucleoside and functions in the same way as I explained earlier with remdesivir. So these are the references which have helped to create this presentation, and my thanks and grateful to all of these scientists who have made these trials happen and published the results. Thank you very much for listening, and thank you.
So, thank you so much, sir, for the wonderful case studies, and hope so everybody is benefited with it. You have done tremendous presentation on the recent literature review and the case studies published for the preventive measures of coronavirus disease. ISF College of Pharmacy is thankful to you for, sir, presenting such a wonderful talk. Thank you so much, sir.
Thank you.
Now, would I like to call Dr. Ramesh Sharma to introduce our next technical session speaker, Dr. Nusrat from PGI.
So very good afternoon to all. So we have our next speaker, Dr. Nusrat. She is a clinical pharmacologist working as a professor in pharmacology, PGI Chandigarh, and she is also in charge of antimicrobial stewardship program of postgraduate institute Chandigarh. She is also uh, uh, hello. Yeah, she's also going to include two different speakers, Dr. Nanda Gamed, uh, MD Pharmacology, and Dr. Praveen Kumar. He's also MD Pharmacology and working in a PGI Chandigarh. Hello? Yeah, please, ma'am, are you able to hear me? Ma'am, you can turn on your microphone. Ma'am, we are able to see your screen, but your microphone is off. Please switch on your microphone. She have left the meeting, so we will try to connect her again.
हेलो हाँ मैडम आपकी आवाज़ नहीं आ रही थी आप फिर से कनेक्ट हो जाइए आप कुछ नहीं बस उसको नहीं एक बार स्क्रीन पे टच करेंगे तो आप अनम्यूट हो जाएंगे
ठीक है मैम यू कैन क्लिक ऑन द स्क्रीन टू ये भी है ठीक है ये भी है साथ में हेलो डॉक्टर नुसरत मिस्टर प्रवीण मिस्टर नंदा you can turn on your microphone there is an icon of microphone in your screen
अपने कर्सर ले जाके चला माइक की तरफ नीचे नीचे लेफ्ट हैंड पे ये अपने मुंह के
when
लेफ्ट पे ना लेफ्ट स्क्रीन पे हाँ जी 395 ये मतलब
when you can click on your microphone so that we can hear your voice दोबारा चल
हेलो ठीक है ठीक है ठीक है अरे ठीक है अभी ठीक हो आप बोलो नहीं करता प्रेजेंटेशन आ गया ठीक है चलिए
प्रजीत प्रताप अदिति श्याम शर्मा बस
आवाज आ रही बता दें
Yes, ma'am. We can hear.
अभी देखो हेलो अभी बात हो रहा है
जी yes
हाँ अब हो गया ना ये देख अभी कुछ भी पता
Okay. So, good morning, everybody. Can you hear me?
yeah. We can hear and we can see the presentation also. You can continue.
Yeah. Good morning, everybody, and I would like everyone to know whosoever is this. I think there is something wrong here. Ooh. Oh God! Again. Please, it is being hacked.
Hey, look at it. Diarrhea. Ooh, it stinks. I wanna give me a spoon so I can